In today's video, I want to dive into what we call Leishmaniasis. And this Leishmaniasis is a protozoan infection caused by hemoflagellates known as Leishmania species. So as we begin, if it is your first time, please remember to subscribe, like the video if you learn, so that you may continue to be notified whenever we release other videos. So we are seeing Leishmaniasis as a protozoan infection which is very fatal, and it commonly also affects immunocompromised patients like HIV, stroke, AIDS patients. And this Leishmaniasis is transported or transmitted by the vector known as phlebotomers. So the vector is the sandfly, which we have drawn here. This is the sandfly, and it has different genera, or what we call genus, whereby we have phlebotomers, genus phlebotomers, and we have also genus Rhizomia or Rhizomia, depending on how you pronounce it. This phlebotomers is found in what we call the Old World. Then Rhizomia is found in the New World. When we talk about New World, we are meaning America. So this one is found in America, American places like Brazil, like South America, and both Central America. So in summary, this is what transmits Leishman species in the New World. Then in the Old World, we could include Africa, Asia, and Europe. So the type of the species, the species is phlebotomers, where we have different phlebotomers species like Sargenti, Martini, Longiparis, Perniciosus. All those are some of the species of phlebotomers which are responsible or acting as the intermediate hosts for these Leishmaniasis parasites. So after understanding these different species which are transmitting, now we need to classify. Leishmaniasis is classified into three, where we have the visceral Leishmaniasis, we have the cutaneous, and the muco-cutaneous. So it is summarized in this table, whereby we are going to look at a different class, whereby we are saying we have the first one as visceral Leishmaniasis. You can call it Karaza. In India, they call it Karaza disease. Karaza meaning blackening, because on healing, this visceral Leishmaniasis leaves black pigmentation, especially on the face and the earlobes, and hence the name Karaza by the Indians. Or you can even call it dam fever. Dam fever. So these are other names for visceral Leishmaniasis. And its name, as suggests, visceral means something internal or inside. So we are saying it is going to affect majorly the internal organs. Those are the reticuloendothelial system organs. So the organs which are affected include reticuloendothelial system organs such as the spleen, the bone marrow, and liver. So these are the three organs which are mostly affected. Sometimes it can continue and affect also the intestines, sometimes the lungs, and the lymph nodes. So lymph nodes, lungs, intestines are also affected, by the primary organs are the reticuloendothelial organs known as the spleen, whereby the spleen is one of the reticuloendothelial organ, we have the bone marrow, and the liver. That is why we call it visceral leishmaniasis. And this visceral leishmaniasis is caused by the Leishmania Donovan complex. So we see it being caused by Leishmania Donovan complex. Leishmania donovani, or sometimes it can be caused by Leishmania infatum. This Leishmania infatum, in America, they regard it as Leishmania chagasi. But in the old world, it is infatum. So if it is in America, we call it Leishmania chagasi, whereas here it is called Leishmania infatum. So they are the same species, but they differ. They are named according to the location where they are found. Then another point is the Leishmania donovani. So these species, two of them, Leishmania donovani and infatum stroke chagasi, are the one responsible for causing visceral leishmaniasis, which affects the spleen, the bone marrow and liver, plus the lymph nodes and other internal organs. And this type, which we are calling kalaza or dam fever, is very fatal when untreated. When untreated, greater than 95% of the patients die when untreated. But after treatment, we have seen it can bring about what we call post-treatment kalaza dermal infection, which we see as black pigmentation of the skin, which we call, the Indians call kalaza disease. Then the second classification is the cutaneous leishmaniasis, which affects majorly the skin, or what we call the dermal layer. So at the site of bite, the infective forms, which we call the promastigotes, they are going to multiply within the macrophages of the skin, which we call the histiocytes. And after multiplying, of course, these promastigotes, when they are engulfed by the macrophages within the skin, they metamorphose into the amastigotes, which amastigotes have ability to undergo binary fission to multiply in number. So when they multiply in numbers within the skin, they lead into what we call the boils or sores, some of them what we call the oriental sores, which cause, so this one affects the cutaneous, or that is the skin, causing oriental sores. And these sores can undergo rupture to cause what we call skin ulcers. This is what we call cutaneous leishmaniasis, and the major cause is Leishmania tropica. We have Leishmania tropica, we have Leishmania major. These are some of the causes. We have Leishmania ethiopica, and sometimes we have Leishmania mexicana, and guyanensis. So these are some of the species of Leishman which are responsible for causing cutaneous leishmaniasis. These first ones, the tropica, the major, and the ethiopica, are responsible for cutaneous leishmaniasis in the old world, the Africa, Asia, and Europe. Whereas Leishmania mexicana, guyanensis, and guyanensis cause in the New World, and they lead to what we call ulceration of the skin, what we call oriental sore. And if it is a dry oriental sore, so dry oriental sore is caused by tropica, whereas wet oriental sores are due to Leishmania major. So in case they ask in exam, which of the following Leishmania species are responsible for causing dry oriental sore, that is Leishmania tropica, whereas wet oriental sore, it is Leishmania major. That is what I want you to note. Then last class, which is number 3, is muco-cutaneous. Muco-cutaneous, this one comes from the word mucus, which means these species target mucous membranes. That is the buccal cavity. Buccal cavity, you can call it mouth, and also it affects the nasopharyngeal parts, nasopharynx parts. So these are the major sites where muco-cutaneous, the mucous membrane within the nasal cavity and the mouth cavity. And the major cause is Leishmania Leishmania blazonensis. Sometimes it can also be Leishmania guyanensis. So these are the major, so these ones belong to the complex known as blazonensis complex, which are responsible for causing muco-cutaneous in the New World. So to the smaller extent, we can also find Mexicana causing this in the New World. So muco-cutaneous form of Leishmaniasis is not common in the Old World, but most commonly in the New World. That is the causes of muco-cutaneous, which affects the buccal cavity and the nasopharynx cavity. So after understanding different classes, but before we leave these classes, we see cutaneous, we can call it daily boil. Daily boils. So if we can ask you which of the following class causes the daily boils, that is cutaneous, which we have called oriental sores, wet and dry. Then mucocutaneous, we can call it spondia. Spondia. This is its other name, which affects majorly the New World. So after understanding the classes and the causes plus the organs which are most affected by different species, now let us dive into what we call the life cycle. And life cycle, we are going to give general, but we shall see how different classes affect different organs in the body. And life cycle occurs both in man, whereby man acts as the definitive host after the sandfly. This is the sandfly inoculating the infective metacyclic promastigotes. So this is what we are calling promastigotes, metacyclic promastigotes, which are going to be inoculated in man when the sandfly is taking a blood meal. So when these promastigotes are inoculated into the skin, they are taken into bloodstream, whereby if it is cutaneous, these promastigotes, they remain within the skin. So the macrophages within the skin are going to engulf these promastigotes, and after engulfing them, they transform into the amastigotes. So if this is the macrophage within the skin for cutaneous leishmaniasis, these metacyclic promastigotes are going to come and metamorphose into the amastigotes. So this is the amastigote formed, and after forming these amastigotes, they undergo division by binary fission to form more amastigotes. So more amastigotes continue to multiply, forming new amastigotes within the macrophages of the skin, that is the cutaneous. And these macrophages can rupture, releasing these amastigotes, whereby these, so we are seeing this having the amastigotes, rupturing, releasing them, and when it releases the amastigotes, they invade new cell, new macrophages. by phagocytosis. So we find others are formed. So when multiple macrophages are affected due to binary fission of the mastigotes, they can swell and rupture and affect new cells, which will come and form a cone within the skin, leading to formation of skin ulcer and sometimes the sores on the skin. That is Pathogenesis. So this one will result into what we call skin ulcerations or ulcers. And these ulcers can, at the end, heal, leaving out scars, leaving out dark scars. That is Pathogenesis. Then when the infective sandfly injects these promastigotes for visceral Leishmaniasis, these promastigotes are going to be transported in the blood and go affect different reticuloendothelial systems. That is the bone marrow, the spleen, and the liver. So we are saying these infective metacyclic promastigotes, after being inoculated, they are transported by blood and they are going to go and be engulfed by the macrophages within the reticuloendothelial system. That is the liver, spleen, and the bone marrow. So after being ingested, the same process will continue. So we are going to see the macrophages within the spleen, within the liver, within the bone marrow, and sometimes within the intestines. They are going to ingest these promastigote and transform into what we call the amastigotes. And we are going to see different amastigotes undergoing binary fission in the macrophages within the spleen, within the liver, those are the Kupffer cells, within the bone marrow. Those are the osteoblasts. So the osteoblasts, the Kupffer cells, and the macrophages of the spleen are going to undergo enlargement. Why? Because there is binary fission, multiplication of the amastigotes by binary fission, which can lead to swelling of the gland. And that's why you are going to see under pathogenesis in the visceral Leishmaniasis, we are going to see conditions like splenomegaly, that is the enlargement of the spleen. We're going to see hepatomegaly, that is the enlargement of the liver cells, and sometimes we are going to see lymphadenopathy. That is enlargement of the lymph nodes, because we said this visceral Leishmaniasis goes ahead to affect also the lymph nodes. So we can find also the lymph nodes swelling. At the same time, since they affect the bone marrow, we can find causing bone marrow aplasia. And this bone marrow aplasia or lack of cellularity of the bone marrow can result into what we call leukopenia. Actually, it can result into pancytopenia, whereby penia means reduced cell line. Pan means several, cyto means cells, and penia means reduced. So you're going to find someone with this visceral Leishmaniasis resulting into both anemia, leukopenia, reduced white blood cell count, and reduced platelet, what we call thrombocytopenia, due to affected organs. And sometimes they can also have diarrhea when the intestines are affected. That is with the pathogenesis of visceral Leishmaniasis, resulting into multiplication of the amastigotes within the macrophages of those organs. And as we said, at the end they rupture, and when they rupture, these amastigotes come and infect new macrophages within these tissues. So you find these amastigotes which continue to multiply by binary fission, form new cells. They continue multiplying, and as they multiply in the cells after being engulfed, they cause what we call swelling of the organs affected. This is multiplication by binary fission. But multiplication is only for the amastigote stage. So this will happen. This is what we can mark about the life cycle in man for visceral and for cutaneous. Then what about muco-cutaneous? With muco-cutaneous, for them we said they affect the mouth parts and the nose and the respiratory part, that is the nasal cavity. So we are going to see the macrophages within the buccal cavity, they are going to be affected. So we are saying it is going to affect the buccal cavity, the nasal cavity, and the pharynx cavity. So these are the sites where muco-cutaneous leishmaniasis affect. And when they multiply, they lead to what we call necrosis of the mucous membrane. We are on muco-cutaneous leishmaniasis. This affects the mucous membranes, that is the mouth and the nasal cavity, leading to tissue necrosis. And this, when there is necrosis, sometimes they can heal when you have good immunity, but sometimes it can progress and causes disfigurement, whereby there is disfigurement of these parts, the mouth and the nasopharynx cavity, and even it can block these components, leading to pneumonia or difficulty in breathing or even difficulty in eating when you have a swelling within the mouth or within the nasal cavity. That is mucocutaneous. But we talked about for cutaneous leishmaniasis. For it, its pathogenesis was causing skin ulcer. Skin ulcers were resulting from the oriental sores, since the mastigotes multiply within the macrophages in the skin. But these ulcers, when they heal, they leave a black scar. And even we saw under visceral, after treatment, they result into what we call post-Kalaza dermal effect due to black pigmentation of the skin. This is after treatment. So this is what we can talk about life cycle in man, who is acting as a definitive host. But the reserve hosts can also be cats, rodents, and other things. The hyenas can act as the reservoir hosts. So in summary, we are seeing the infective phlebotomous fly or rhizomia ingesting or inoculating metacyclic promastigote, which is the infective stage. These promastigotes are engulfed by the macrophages within different organs, that is the skin for cutaneous, mucous membrane for mucocutaneous, and internal organs for the visceral. And those macrophages, after engulfing the promastigotes, they transform them into the amastigotes, which undergo multiplication by binary fission, leading to the swelling of the organs which are affected. And some macrophages rupture, releasing the amastigotes, which go and infect other new macrophages in the sites where it is affected. And this one results into these pathogenic features, pathogenesis, or clinical symptoms due to that multiplication of the amastigotes within the macrophages. So the life cycle will continue when the sandfly, female sandfly, is taking a blood meal from infected person. So when this man is infected with the amastigotes within the macrophages of different organs, this sandfly is going to ingest these amastigotes. And these amastigotes are found in the macrophages. So we see the macrophages containing amastigotes being ingested by the sandfly, and they go into the gut. So when these macrophages reach in the gut containing the amastigotes, due to change in the temperature, That is, decrease the temperature, because we know in man temperatures are high, 37 degrees. But in the sandfly, temperature tends to reduce. So due to decrease in temperature and increase in pH, these two conditions are going to trigger, after digestion of the macrophages, these mastigotes are released. And after releasing these mastigotes from the macrophages by digestion within the gut of the sandfly, these mastigotes transform into procyclic promastigotes. They're going to transform into flagellated promastigotes. That is what is causing metamorphosis, is decrease in temperature and increase in pH, which triggers metamorphosis of the mastigote, which are released from the macrophages which were ingested by the sandfly, and we form procyclic promastigotes. And these procyclic promastigotes are going to undergo binary fission, whereby one promastigote is going to form two promastigotes. So that is what we are calling binary fission, which occurs within the gut of the sandfly, or Phlebotomus or Rhizomia in the New World. So after undergoing different multiple binary fission, we form multiple promastigotes. And these promastigotes, within 10 days, they are going to go and invade the mouth parts. So these promastigotes are going to migrate towards the mouth parts of the sandfly, that is the proboscis. So they migrate to the proboscis, not the salivary glands, like the trypanosomes. These ones, they go to the proboscis of this sandfly, ready to be inoculated when the sandfly is taking a blood meal. So these promastigotes, which we are going to call metacyclic promastigotes, which are found within the proboscis, which is the mouth part of the sandfly, are ingested, inoculated in man when the sandfly is taking a blood meal, and life cycle will continue. So this is the life cycle in a vector, and this is the life cycle in man. So this is what we can talk about. Please, as a student, don't forget that the infective stage is metacyclic promastigote. The diagnostic stage in macrophage is the mastigote. But then in tissue culture, it is promastigote. So both mastigote and promastigote can be diagnostic depending on the method. If it is culture, we find the promastigote. But if we are examining the macrophages, we find the mastigotes, which are intracellular. We normally see them inside the macrophages of the tissue biopsy or the blood or the bone marrow aspirate, which is being used as a diagnostic marker. The signs and symptoms can be due to the organ infected, the blackening of the skin for cutaneous and presence of skin ulcer, then lesions and necrosis within the mucous membrane, which can result into difficulty in breathing and eating due to the presence of inflammatory tissues within the mucous membrane. Then we are seeing organomegaly, which we can also see as a sign and symptom. Apart from these, the mild ones, we have intermittent fever. You find someone who has a fever which is on and off, can also be. Then there is cachexia, there is fatigue. Then we have seen anemia in case of visceral Leishmaniasis, also results in 2. So this is what we can summarize in this session. Hope you have learned something in this session. Please remember to like the video and share to many others so that we can continue to grow. Thank you so much. In the next video, we shall look at the lab diagnosis of Leishmaniasis. May God bless you.